Okay, so this video is going to be covering the second half of Module 8 on Mendelian genetics. So last time we talked about genotypes, which we said were the two-letter designation for a genetic trait. So it would might look like this, or like this, or like this. And then we talked about phenotypes, which are the actually the observable characteristic or trait that is based on the genotype that you have. We talked about homozygous genotypes. And homozygous genotype will be where the two-letter designation, the two letters are the same. And then we talked about heterozygous genotypes. And that would be one in which the two letters in the genotype are different. Okay, so it would be that. Okay, and then we looked at Punnett squares and how Punnett squares can be used to predict the genetic characteristics, both genotypes and phenotypes for offspring. And when we did our Punnett squares, we just did monohybrid crosses where we're just interested in, we just look at one trait at a time. So let's run through an example of one of those really quickly. So let's say that tongue rolling, being able to roll your tongue, is a dominant trait. So if you have one dominant letter in your genotype, you will be able to roll your tongue. So let's say that we have a heterozygous tongue roller, and that heterozygous tongue roller is married to a heterozygous tongue roller. What are the possibilities and percentages for their offspring? So a heterozygous tongue roller, since we said tongue rolling is dominant, then since they're heterozygous, they would both look like that. So we can draw our Punnett square, and we will put doesn't matter, one across the top and the other parent across the bottom, and then we just match up the letters. So this one would be that, this and this, this and this, and then lastly this. So if we look at the genotypes, what are the possibilities for genotypes? So you can have RR, and that's 1 out of 4, or that's 25%. Would have that genotype. We see that 2 out of 4 have this genotype, so that would be 50%. And then the other genotype is this one. So again, that's 1 out of 4. So that's 25%. And those percentages should add up to 100. Okay, so that's the genotype. 50% would be heterozygous. 50% would be homozygous with the dominant trait, and the other 25% would be homozygous with the recessive trait. Okay, then what about the phenotypes? What would the phenotypes look like what are you going to observe? Well, as long as you have one of the dominant alleles, you're going to be able to roll your tongue. So 1, 2, 3 out of 4, or 75%, would be tongue rollers. And 1 out of 4, this one, would, because that person just has the recessive alleles, 1 out of 4, or 25%, would be a non-tongue roller. Now remember, we said last week that these are just, these are probabilities, so it doesn't mean that if this couple were to have 4 children that everything would turn out exactly like that, because which genotypes get matched up depends on chance. And so, but if you were to look at a large enough sample, generally these percentages would hold true. Okay, so that's a monohybrid cross, where you're just looking at one trait at a time. Okay, now we're going to look at dihybrid crosses. And a dihybrid cross is one in which we look at more than one trait at a time. So we're a dihybrid cross, you look at 2 traits at a time. So let's say that we are interested in looking at, so let's look at plants, and we are interested in looking at height, and also color. Okay, so 2 traits that we will look at the time. So what if we have a plant that has these, it's tall, it's homozygous tall and homozygous purple, and we're going to cross that with a homozygous recessive, homozygous short, and homozygous white. Okay, so what you have to do for a dihybrid cross is you have to look at what are the possible ways within each organism that the alleles could be arranged. Okay, so if we look at this one first, the only options are that a big T could be paired with a little t, right? Because they're both big. They're both big P's. They're both capital letters. So a big T could be paired with a big P, and a big T could be paired with a big P. So there's just one possibility. So for this particular plant, that's the only possibility. So on our Punnett square, we would put that across the top, and then we look at the other organism, and again, the only options are that a lowercase t could be paired with a lowercase p. Okay, so we'll put that one, I need to make that more clear that it's a lowercase. Okay, so that's lowercase. So then we'll put this genotype along the other side, because that's the only possibility for that particular organism. And then we just draw our Punnett square. So for this particular mating, sorry, this should have gone like this, because we're looking at two traits at a time, so that we put the 2 alleles, put the things next to each other. So the only possibility for offspring for this kind of mating is that a capital T and a lowercase t, and a uppercase P and a lowercase p. So if we cross these two types, so a tall purple and a short white, 100% of the offspring will have this genotype, and 100% of the offspring would be tall purple plants. Okay, tall plants with purple flowers. Okay, so that's an easy one where the combination of letters is pretty straightforward because within the two genotypes there's only one possibility. Okay, so then what happens? Let's do one that's a little more complicated. What if we cross these two plants? So we're going to cross that with another heterozygous. So it's heterozygous for tall, heterozygous for purple, heterozygous for tall, heterozygous for purple. Okay, so then what we have to do when we draw our Punnett squares is we have to look at what are the possibilities, how can these alleles match up? Okay, so first of all, the uppercase T could go with the uppercase P. So we put that one. And then the uppercase T could be with the lowercase p. Okay. Then the lowercase t could be with the uppercase p. And lastly, the two lower cases could be together. Okay, and then you would do the same thing for the other plant. It's going to be the same because the genotypes are exactly the same. But so the uppercase T, both the uppercases could go together. The lowercase t could go with the uppercase P. Well, actually, let me do that differently. So the uppercase T could go with the lowercase p. And then lowercase with uppercase. And lastly, the two lower cases could go together. Okay, then we draw our lines for our Punnett square. So we're going to go across there, and then down in between each of these. Okay, and then what do we do? Then we just match up. So this would be uppercase T's and both uppercase P's. This would be two uppercase, and this would be that and that. This would be uppercase, lowercase, uppercase, uppercase, and this would be that, 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 and that. Okay, so I'm going to go ahead and finish filling this out. Okay, so those are the possibilities for the genotypes from this mating of a heterozygous tall purple with a heterozygous tall purple. Okay, so then what you have to do is you have to go through and look at the, in this case, 16 genotypes and figure out what would the phenotypes be for them. Okay, so I'm going to erase this up here. It's going to be tough because we're going to run out of room, but so this one right here that I'm pointing to would be tall and purple. Okay, so that's one possibility, is they could be tall purple plants. So let's go through and count how many would be tall and purple. Okay, so this would be tall and purple. This would be tall and purple. This would be tall and purple. Tall and purple. Tall purple. Tall and purple. Tall and purple. This is tall purple. Tall and purple. That's it. So 1, 2, 3, 1, 2, 3, 4, 5, 6, 7, 8 would be tall and purple. One, 2, 3, 4, 5, 6, 7, I missed one, 8. So 9 out of 16 would be tall and purple. Okay, then what other possibilities do we have? We can have tall and white. So let's look for tall and white. So to be tall and white, they would need to have at least one uppercase T, but both lowercase p's. Okay, so this would be, I'm going to change colors here. So tall and white would be this one, this one, this one. Okay, so 3 of them would be tall and white. Oh. So 3 out of 16 would be tall and white. Okay, now let's go to red. And now what's another possibility? They could be short and purple. Okay, so the short and purples would have lowercase t's and at least one uppercase p. So 1 short and purple, 2 short and purple, and 3 short and purple. So 3 out of 16 is short and purple. And then lastly, we'll go back to black. The last ones is that they could be short and white. And that means all the letters are lowercase and we have only one of those. So 1 out of 16 should be 1 out of 16, 3 out of 16. So 1 out of 16 will be short and white. So that should add up to 9, 10, 11, 12, 13, 14, 15, 16. So of the offspring, the probability is that 9 out of 16 of them will be tall and purple, 3 out of 16 would be tall and white, short and purple would be 3 out of 16, and short and white would be 1 out of 16. Okay, so that's it's a little more complicated, but and you have to that's how you would do a dihybrid cross, is look at each one and figure out what the possible genotypes are, and then figure out the percentages. Okay. And you could do that with any number of traits. Let's do another one real quickly. What if we had a homozygous taster? So here we're going to look at there are certain chemicals that some people can taste and some cannot. And being able to taste is a dominant gene. So this is a taster, and the person can roll their tongue. So they're a taster and a roller, and we're going to cross them with a non-taster and non-roller. Okay, so they can't roll their tongue and they don't taste the paper. Okay, so when we look at our Punnett square, we look at this person first. The only way these can match up is you can have a capital T with a capital R. That's the only possibility. Okay, and over here, the only possibility is a lowercase t and a lowercase r. So then we draw our Punnett square, and all of the offspring will look like this. So 100% of the offspring would be tasters and rollers, and they would be heterozygous for each of those traits. Okay, so that is an example of that's another example of a dihybrid cross where you look at two characteristics at once instead of looking at just one characteristic. Okay, now we are going to start looking at how genetics is related to sex or gender, and how traits can get passed on differently depending on gender. So let me remind you, first of all, that humans, so we're going to concentrate on humans now, have 23 pairs of chromosomes. And 22 of those pairs are called autosomal. So 22 pairs are autosomal. And that simply means that they do not determine the sex of an individual. So they can determine hundreds or thousands of other characteristics, but they do not determine the sex. And then one pair of those is a sex chromosome, and the sex chromosomes do determine the sex of an individual. So 22 pairs of these 23 are autosomal, and one pair is the sex chromosome that does determine the sex. Okay, so in a female, for a female, during meiosis, when the gametes are produced, we use the letters X and Y. And in a female, both of the chromosomes are X chromosomes. And in a male, one of the chromosomes is an X, but the other chromosome is a Y. So in meiosis, when meiosis happens and we take that one cell and we produce cells that have just, half of that chromosome. In a female, they can only be X's. Whereas in a male, when this chromosome splits and you get the 4 gametes produced, half of them will be X's and the other half will be Y. Okay, so if this is on the chromosome, those are going to split. Well, then all of the ones, the only thing that a female has for the gamete to be is an X, whereas for a male, the gamete could be an X or a Y. Okay, so that happens during meiosis. So then we look at what are the probability of the offspring. Okay, so if we put the female on top. Again, it doesn't matter how you do it. You could put the male on top too. It doesn't matter. And then we put the chromosomes or the alleles for the male on the side, and we draw our Punnett square. And we match these up. So 1, these are all X's. An X and an X would go here. An X and a Y would go here. And another X and a Y would go here. Okay, so then what does this show us? Well, it shows us that shouldn't have made those lines so long. So when a male and female have children, these are what the probability of their offspring are. And so you can see that 50% or half, 2 out of 4, would be XX. So those are girls. And 2 out of 4 or 50% would be XY. Those are boys. Okay, so every time a couple has a child, there is a 50% chance that it will be a girl and a 50% chance that it will be a boy. And again, these are probabilities, so it's not going to come out exactly like that in every family. But if you were to look at, a couple hundred or 1,000 offspring of lots of couples, worldwide, it does come out that about 50% are girls and 50% of boys are conceived. Okay, so that's how the sex of offspring is determined, is on the sex chromosomes. With the female being XX and the male being XY. Okay, there are some traits that are more common in particularly in males than in females. And we call these traits sex-linked traits. Okay, so one example, one of the organisms that is used a lot to study genetics is Drosophila, which is fruit flies. Partly because there are some genetic characteristics of fruit flies that are easy to study, and also partly because fruit flies reproduce really rapidly, so you can look at multiple generations in a relatively short period of time, because they're able to reproduce every 8 hours. And so within a 24-hour period, you could get 4 generations of fruit flies and look at the characteristics. So one characteristic in fruit flies that we look at is the eye color. And what happens is that for these sex-linked traits, some of them have alleles that can only exist on the X chromosome. So remember, so this is a female and this is a male. Well, there are some traits that can only exist on the X chromosome, and the reason for that is that the Y chromosome is smaller than the X chromosome. So if we were to look at a pair of X chromosomes, they would look like that. So for a female, the chromosomes would look like that. For a male, the X would be that size, but then the Y chromosome is smaller. So because the Y chromosome is smaller, it can't carry as many alleles or as many genes on it. So because of that, there are some traits that can only exist on the X chromosome. And in fruit flies, one of those is eye color. So and in particular, we're going to look at the red eye color. And the red eye color is dominant. So let's say we have a heterozygous red-eyed female. Okay, so that female would be this and this. And then what if we had a red-eyed male, and we're going to mate these two fruit flies. Well, since we just said that in a male, the trait for eye color is only carried on the X chromosome. It's not carried on the Y. So this male would just have a single R. And the way we write these to designate that it's a sex-linked trait is that we write it like this. So for the heterozygous female, the female has two X chromosomes, right? And since she's heterozygous, one of those would be a capital R and the other would be a lowercase r. For the male, so a male is this and a Y. Well, if he's a red-eyed male, then on the X chromosome he would have the dominant gene, but he would have nothing on the Y because eye color is not carried on the Y. Okay, so then when we do the Punnett square for this crossing, what we would do is, okay, here's how we would write it. So the female would be this. And the male would be an X and a Y. An X. Yeah, X and a Y, sorry. And the male would have the dominant allele on the X, but he would have nothing on the Y. Okay, so then we can draw the Punnett square to determine what the offspring would look like. Okay, so then we look at the offspring, And this would be, again, we're just going to match them up. So a capital X with a uppercase R and another capital X with an uppercase R. This would be uppercase, or two X's, and one of those would have the uppercase and the other would have the lowercase. Okay, then this would be an X and a Y, and the X has the uppercase R and the Y has nothing on it. And then this one would be, the X has the lowercase r and the other one would have a Y. Okay, so then if we look, so now we look at these, and again we see 50% are females, so these are the females here, and here are females, and the bottom ones are males because they have the X and Y chromosome. Okay, so if we look at the females, so we look at this upper layer, 100% of the females have a red eye. Okay, 100% of the females are red-eyed. So these are red-eyed and these are red-eyed because they each have at least one of the dominant allele. But then so these are females. If we look at the males, this male, as long as you have one of the dominant alleles, you would have red eyes. So that you would have 1 out of the 2, or 50% of the males would be red-eyed. And the other 50%, since they just had a recessive allele, they would not have red eyes. So 50% would have non-red eyes. Okay, so you'll get different percentages of the traits in males and of the expression of the trait in males and females because it's just carried on the X chromosome. Okay, so that is an example of a sex-linked trait. And one sex-linked trait that maybe you have heard of is called hemophilia. Hemophilia is a genetic disease in which blood does not clot properly. So these individuals who have hemophilia have to be very careful about getting any kinds of cuts or lacerations because their blood doesn't clot properly, and then they have to be given clotting factors that aren't present in their blood so that the blood will clot. So hemophilia is a sex-linked trait. And interestingly enough, this is a trait that exists in the in English and Russian royalty, and we're going to look at how that can affect the offspring in a family if hemophilia is present. Okay, so if we cross a heterozygous, hemophilia, let me say first of all, hemophilia is it's a recessive trait. So it's recessive and it's sex-linked. Okay, so if you're a female, the only way that you could have hemophilia is if you're on both X chromosomes, there is a lowercase r. Then this would be a hemophiliac female, this would be a non-hemophiliac female. As remember, to have since it's a recessive trait, you have to have 2 of the alleles if you're a female. So non-hemophiliac female. For a male, so a male has the X and Y chromosome. If the male looked like this, that would be a non-homophiliac male. And this would be a hemophiliac male. Because since it's a sex-linked trait, you only need to have 1 of those on the X chromosome for it to be expressed. So even though this male has only 1 of the recessive genes, since it's a sex-linked trait, that's all you need. Okay, so let's see what would happen if we had a heterozygous non-hemophiliac female. So heterozygous, so she doesn't have the disease, but she's heterozygous, and that person is going to marry a non-hemophiliac male. Okay, so there's no gene for this is on the Y chromosome, it's only on the X chromosome. So this is a heterozygous female, and this is a homozygous male, but neither one of them have hemophilia. Okay, so non-hemophiliac, and this one also is a non-hemophiliac. Now, what we would say about this female is we would say, even though she is a non-hemophiliac, we would say she is a carrier for hemophilia because she has this trait. Now, we don't use the word carrier for traits that are on autosomal chromosomes. We only use it for traits that are carried on the X chromosome. Okay, so even though she doesn't have hemophilia, she carries the gene for it and could pass it on to offspring. Okay, so let's look at then what would the Punnett square look like. Okay, so for the female, these would be her 2 alleles, and for the male did not have hemophilia. Remember, it's a recessive trait, and there would be nothing on the Y chromosome. Okay, so then we can draw our Punnett square, and we can look at what the offspring would look like. So this is 2 X's, and this would be an uppercase and an uppercase. Okay, so that would be a non-hemophiliac. This again, 2 X's, and an uppercase would be on one of those, and a lowercase would be on the other. Then we look at the bottom row, so it's an X and a Y, and the X would have the uppercase H, and nothing is carried on the Y. And this one would be another male, X and Y, but here the X has a lowercase h. Okay, so then we look at the offspring, and we look at the probabilities for the offspring. So the top row are going to be the females. So half of the females would be both uppercases. Half of the females would be uppercase, lowercase, but there are 0% hemophiliac. Because for a female to be hemophiliac, the female has to have both of the recessive alleles. So the 0% are hemophiliacs in the females. Then we look at the males, and we see that half, this would be a hemophiliac because, oh, I'm sorry, this is a non-hemophiliac. This would be a hemophiliac because in a sex-linked trait, the male only needs to have one of the disease-causing allele to have the disease. So 50% of the males, half of the males would be hemophiliacs. Okay, so sex-linked traits are they're more common among male offspring because in order to have the disease, you only need to have one of the alleles. Okay, so that is hemophilia, and how hemophilia gets passed on through, you can have a woman and a male, neither one of which have hemophilia, but they can have hemophiliac male offspring because you only need to have one of those alleles in order for the disease to be present. I mentioned last week that there are some traits that are not dependent on just one gene, but they are dependent on the interaction between several genes. So this just means that traits can be determined by interaction of several genes. So eye color is an example. Your eye color is actually dependent on one gene that is on chromosome 15, and then another gene that is on chromosome 19. So it's not a single gene that determines your eye color, but it's different genes. And we know that height and skin color are other traits that rely on the interaction of more than one chromosome, and that's called poly. So poly means like many or more than one, polygenetic inheritance. Another thing that I mentioned briefly last week is something called incomplete dominance. And incomplete dominance is where you can have different traits, but neither one is dominant. So if we're looking at flower color, the dominant, we'll call the dominant allele R and the recessive allele R. So if you have this genotype, you would be red. If you have this genotype, you would be white. But since it's incomplete dominance, if the plant is heterozygous, this would be pink. Okay, so it's something that is somewhere in between. Okay, one trait that is interesting to look at is blood type. what blood type you are. So there are 4 possibilities for blood type, and those 4 blood types are type A, type B, type AB, and type O. And what we know about blood type is that O is recessive to A and to B. So what that means is that if you have, if you're 2 alleles, so you could have an A allele and an O allele. Since O is recessive to A, this would be type A blood. And if you had this, if your 2 alleles were this, so if this were your genotype, since O is recessive to B, this would be type B blood. Okay, so we know that O is recessive to A and B. So how could you have, what are ways that you could have type A blood? Okay, so for type A blood, your 2 alleles could be this. You could get an A from each of your parents, or it could be this. We got an A from one parent and an O from the other parent. For type B blood, you could have both. You could get the B allele from both parents, or you could have gotten a B from one and the O from the other, but you would still have type B blood because B is dominant over the O. Okay, but then we also know that A and B are not dominant over each other. So if from one parent you get an A allele and you get a B allele from the other parent, then this is type AB blood. Okay, because neither A and B are not dominant over each other. And then the only way you can have O blood is that both of your alleles have to be O, because O is the recessive one. Okay, so that is how blood type works. In this case, when we say that if you have an A and a B, then you have type AB blood, what we call that is we call that codominance. So neither one is, you don't get something in between. They both get expressed in a different type of blood type called type AB. Okay, so let's look at a Punnett square. What if we have one parent has this genotype for blood type and the other parent has this genotype? What would the offspring look like? Okay, so we're going to put these 2 alleles on the top, and we'll put the other 2 alleles on the side, and then we'll draw our Punnett square and see what the offspring would look like. So this one would be AA, this would be AO, this would be AB, and this would be BO. Okay, so then if we look at the blood types, this is type A and this is type A. So 2 out of 4, or 50%, is type A. This is type AB, so 1 out of 4, or 25%, is type AB. And then 1 out of 4, again 25%, is type B. Oh, type B, because this is type B. Okay, so when the blood is tested, you wouldn't necessarily know what the genotype is by just testing the blood, but this would be type A. But these both would be type A blood. This would be type B, and that would be, I'm sorry, this is type AB, and this is type BO. Okay, let's do another one of those. What if a couple who have this, one of the pair has that genotype and the other pair has this genotype, what would the offspring look like? Okay, so we're going to put the B and the O across the top, and we'll put the A and the O across there, and then we draw our Punnett square. I don't know why I keep drawing these lines so long. So we'll draw that there. And then we can look at the offspring. So this would be AB. This would be A and O. Put these two together would be B and O, and this would be O, O. So if we look at the offspring, 1 out of 4 would be AB. 1 out of 4 would be AO. 1 out of 4 would be BO. And 1 out of 4 would be OO. So this is type AB. This is type A blood, because the A is dominant over the O. This would be type B, because B is dominant over O, and this would be type O. Okay, so here's that example of incomplete, or of codominance. Both the A and the B get expressed so that they have type AB blood. I'll give you a problem to do here. I have type O blood, and my husband, we don't know what he has, but we know that some of our children have type A blood, and some of our children have type O blood. Okay, so what that should tell you, if some of our children have type A blood, that means my husband must have an A allele, because I don't have an A allele to give. I only have O's to give. So if we draw a Punnett square, I would look like this. Okay, those are my 2 alleles. If some of our children have type A blood, so I can only give the O. Okay, that's to every child I can only give an O. So if some of our children have type A blood, that means my husband has to give an A, right? But if some of them have type O, that means he also has to have an O to give. So even though we don't know his blood, he has never had his blood type determined, we can look at our offspring and see that because some of our children have type A blood and some have type O, this has to be what my husband's genotype is, because that's the only way that that we could have some children with type A and some children with type O blood. Okay, I hope that makes sense. Some traits that maybe you have heard of. There are some people who can't tolerate lactose. Lactose is the sugar that is in milk. And so some of these people, they might need to drink milk alternatives, like that don't have lactose in, where you can buy milk in which the lactose has been removed. The lactose intolerant, is it's a trait that it's not carried on a sex chromosome. It's carried on an autosomal chromosome, and so a person, we could say that a person with this genotype, and it's a recessive trait. So we would say that this person carries the trait but doesn't have it, because in order to have it, you would have to have the two lowercase l's. There is another genetic condition that is called Huntington's disease. And this is a disease that is not carried on a sex chromosome. It's carried on an autosomal. And it is it's a dominant trait. It's dominant, and it's a fairly rare condition, which tells us that most people are recessive, because it's not a very common trait. So most people are, if we use the letter H, most people would have that. But and then if in order to have the disease, you would have to have this, or you would have to have this, because it's not in complete dominance. So as long as you have one of these, you would not have the disease. So here's what can happen if you have a person who has the disease. So let's say this person is heterozygous, and they have the disease. So they would be this. And if they marry someone who does not have the disease, that person would be this. So then if we look at the Punnett square, make the Punnett square. So this would be capital H, lowercase h, capital H, lowercase, 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 lowercase. So remember we said that it's a dominant trait. So most people are this. Most people do not have the disease. But if somebody who has Huntington's marries somebody who does not have Huntington's, there is a 50% chance that their offspring will have Huntington's. So 50% will be normal, and 50% will have Huntington's. Now, the problem with Huntington's disease is that it does not show up until the symptoms of Huntington's disease do not appear until a person is in their 40s or 50s, which for most people, that is the time after they have already had children. So they might be passing on, if somebody has Huntington's disease and they have already had their children before they learn that they have that, before the symptoms show up, they could be passing on Huntington's disease to their offspring without knowing it, because they didn't know they had it until after they were finished bearing children. There's no cure for it. It's a progressive neurological disease that ultimately ends up being fatal. But symptoms don't appear until you are in your 40s or 50s. There's a it's a fictional book. I think it's called something like Inside the O'Briens, which is about a New York firefighter who began to develop symptoms of Huntington's disease when he was in his 50s. And by that time, he and his wife, I think they had already had 3 or 4 children, and their children were, most of them were near or already, all of them were either near being adults or they were adults. And so the book deals with the idea of whether because now with genetic testing, because the father tested positive for it, that meant that, because the father had it, that means there was a 50% chance that his children would have it. And now because we can do some genetic testing, the children could find out whether they were going to develop it. And so the book is about whether these children should find out, because at least one, maybe 2 of the children were married themselves and had not yet had children. So then they kind of wrestle with this idea about should they find out so that they know the risk of passing it on to their own children. So if they have it, would they want to risk passing it on? Would they want to risk having children and passing it on to the next generation, knowing that it is ultimately a fatal disease, and yet you can live 40 to 50 years before you start to develop symptoms, and you can live into your 60s or even 70s with the disease, depending on when those symptoms appear. So with the genetic testing that we have available now, there are some kind of ethical issues that we have to, people have to kind of wrestle through, and what kind of decisions do you want to make based on genetic information that might be available to you. I will mention one other condition, and then we'll stop for today. You may have heard of a condition that's called Down syndrome. Down syndrome children they tend, usually they have some degree of mental retardation. They will have, often will have heart defects. Usually those heart defects, many of them can be repaired surgically, but they have some degrees of mental retardation. They tend to be short. Their muscles, they don't have a lot of tone in their muscles. Their hands tend to be kind of short and thick fingers. And I think your book gives a figure of about 1 in 1,000 babies are born with Down syndrome. Down syndrome does tend to increase with the age of the mother. It's still a very small percentage, but the age of the mother can affect it. And Down syndrome the trait can be passed, can be either from the father or the mother. So it doesn't just come through the mother. And what Down syndrome is caused by an extra chromosome. So the term that is used is trisomy 21. So remember that there are 23 pairs of chromosomes, and we number those. So we would say pair 1, actually they come in pairs. So this would be pair 2, this would be pair 3. Well, in Down syndrome, the pair that is called 21, instead of having only 2 chromosomes, it has an extra chromosome. And that extra chromosome is what causes the the characteristics of a Down syndrome child. And as I said, it could either be the sperm that has an extra chromosome, or it could be the egg. But it's just the extra chromosome is what leads to the characteristics of Down syndrome. Another thing that so this would happen during meiosis, when the egg and the sperm are going from being diploid cells to haploid cells, if you end up with that extra chromosome. Another thing that can happen in meiosis is that you can get what is called a mutation. And a mutation is just that one of the alleles changes, usually pretty drastically. So an allele radically changes during meiosis. And there is a condition that's called Hutchinson-Gilford progeria syndrome. And that syndrome is what you get is premature aging. So these children age at a very, very rapid rate. So that by the time they're like 4 years old, they look like an old person. They have a lot of wrinkles. Their bones are weak. They have, arthritis. They're losing their hair. The kinds of things that happen with aging happen by about age 4. And this condition is caused by a mutation. So there's nothing wrong with the egg or the sperm of the mother or father, but there's a change that happens actually during meiosis that leads to this condition. Okay, so mutation is another way that a genetic abnormality can be caused, is through changes that actually happen during meiosis. I think we are going to stop there. So you should be able to do Punnett squares for monohybrid and dihybrid crosses, and you should also be able to do Punnett squares for traits that are carried on sex-linked chromosomes, those that are traits that are carried on the X chromosome and not on the Y. Thank you.